Good morning, everyone, and welcome to our last edition of these Conda Lab Talks. Um, well, we are at the end of the year, so thank you for attending to today's webinar. We will talk about one or even perhaps the best known pathogen in the world of food safety, that is Salmonella. My name is Aletia Cruz. I am a project manager for microbiology here at CondaLab. So without any further ado, let's start with the presentation. So this presentation is going to be divided into 5 parts. We will start with a quick review of Salmonella and Salmonellosis. Then we will continue to the monitorization and control of these bacteria in the European Union. We will go into the detailed revision of ISO 6579 Part 1 with its amendment in 2020. And then to close, we will talk about something very exciting that's coming. That is a new alternative method for Conda, from Conda, that is Condagene, and we will end with the available products at the moment. So without any further ado, let's start with Salmonella, the pathogen. From the end of the 19th century, Salmonella began to be documented as a source of disease. And in fact, this bacterium was named after Daniel Elmer Salmon, a veterinarian of this time, although he was not the one who discovered this microorganism. That should be noted. Salmonella belongs to the Enterobacteriaceae family. That is, it is a facultative anaerobic, gram-negative, predominantly mobile, usually with the aid of peritrichous flagella, that is the ones around the cell body. They produce gas from glucose and use citrate as their only carbon source. Today, the genus of this bacillus is classified into 2 species, Salmonella bongori and Salmonella enterica. This latter is further divided into 6 subspecies and more than 2,000 serotypes, at least 100 of these included in the enterica species and subspecies, that is enterica enterica. And they are defined on the basis of somatic O and flagellar H antigens. So it is not surprising that their taxonomy sometimes can create confusion. The full name of a serobar would be, for example, Salmonella enterica enterica, serotype Typhimurium, or simply and more commonly referred as Salmonella typhi. Biochemical assays of the O and H antigens are used to characterize these different serobars. The worrying thing is that the most important serotypes are not limited to a single species or host. And boy, does Salmonella have a great variety of hosts, from animals such as cattle, pigs, birds, reptiles, rodents, to plants, and of course, human beings. Another factor that contributes is that this great variety of serotypes are not clearly identified. Many are what we know as wild serobars, and the diversity of sources makes it difficult for the prevention programs to be implemented both in each country and also locally. The most common current approach consists of measures to achieve a minimum of infections with serotypes important to public health, such as Salmonella enteritidis, or Salmonella typhi, or Salmonella heidelberg, without, of course, neglecting the control of emerging serobars. Although all serotypes are capable of causing disease in humans, some are specific and may reside in one or a few animal species, such as the case of Salmonella dublin in cattle or Salmonella choleraeus in pigs. Another classification, an important one, is historical, since Salmonella has been clinically differentiated as invasive or non-invasive, that is, typhoidal or non-typhoidal. Through typhoidal Salmonella, it produces what we know as typhoid fever, on which we will not focus today. On the other hand, after the infection of non-typhoidal Salmonella serotypes, the disease developed has a name that we know all as salmonellosis. This commonly occurs from eating contaminated food. Usually, these non-invasive serotypes cause gastroenteritis, and it really can pass without much complication and treatment, but of course, there are, as usual, at-risk populations in which the disease can be complicated and can be even fatal. The most common symptoms are abdominal pain, vomiting, diarrhea, cramps, and fever, but of course there may be other more serious complications. The usual treatment is electrolyte replacement and rehydration. And in general, although we may not believe it, antibiotic treatment is not recommended in mild and moderate cases because they may not kill the bacteria completely and in turn can be selective for resistant strains, rendering, of course, the drug ineffective and dangerous. Salmonella can attack both healthy hosts and hosts with pre-existing conditions, and in addition to gastroenteritis, cause bacteremia, meningitis, and tonsil infections. In the case that there is a spread of the bacteria to other parts of the body, the treatment is aggressive with antibiotics, but with caution. This has to be done according to the local surveillance system regarding the resistance of microorganisms. So Salmonella is widely distributed in domesticated and wild animals, often food animals such as poultry, pigs, cattle, as we've mentioned, and also pets, including cats and dogs and reptiles, even turtles. And in fact, a small part of the cases includes direct transmission from infected animals to humans. In other words, Salmonella has the ability to traverse the food chain from start to finish, from animal feed through primary production to our homes or to food service establishments. The most common way of contracting this bacterium is through contaminated food or of animal origin, but now it has also been found commonly in vegetables due to sometimes the contamination of unsuitable water. A CDC report suggested that nearly half of the illnesses and 23% of the deaths were associated with agricultural products. And this occurs because Salmonella is a natural inhabitant of the gastrointestinal tract of these hosts. But once outside of this habitat, it can be transported to surface waters, surviving pH and temperature changes, even the UV radiation from sunlight, and the availability of few nutrients. It is also capable of penetrating the stomata of plant leaves and roots and also attaching to the surface of these fresh foods that provide a niche for these bacteria because of Salmonella's ability to generate biofilms. So in these vegetable produce stands, there are some phytopathogenic bacteria that can help the development of Salmonella as well by sometimes macerating the plant tissue such as Pectobacterium, while there are of course other species that can inhibit or even eliminate Salmonella. And more dangerously, contaminated plants do not show any signs of deterioration, so it is impossible to tell with the naked eye if it's safe to consume or not. In recent years, the number of outbreaks due to these non-traditional sources, such as fruits, vegetables, or spices, has increased considerably due to the above and added to the fact that Salmonella is able to survive for long periods in products with low moisture content. This, of course, is coupled with the trend of the last decade to promote a healthy lifestyle and with the consumption of fruits and vegetables, little processed or even raw. Even more international trade allows the introduction of new strains, which adds one more variable to the problem. Also, there is risk of cross-contamination and recontamination affecting mostly prepared foods, and where Salmonella cells spread from one area to another, and there are additional factors that allow this persistence, such as an inadequate storage temperature, undercooking, or even poor food handling. So in a report on the global burden of invasive non-typhoidal Salmonella, it was stated that even in the 21st century, this pathogen continues to be a major cause of illness and death. The Salmonella species is estimated to cause almost 94 million cases of gastroenteritis worldwide annually, with 155,000 deaths. The main serotypes that cause this disease, as we have seen, are Salmonella typhi and Salmonella enteritidis, which we can see on this table on the left. On the other hand, according to WHO data, the burden of foodborne diseases is considerable, since every year 1 in 10 people fall ill and 33 million years of healthy life are lost. On the other hand, diarrheal diseases are the most common and the result of unsafe food, with which more than 500 million people fall ill annually, of which more than 200 of these million are children under 5 years of age, and a quarter of all of these numbers are caused by Salmonella. Now, in this second part, we will review the European Union policies regarding Salmonella. So in the EU, salmonellosis is the second most frequently reported gastrointestinal infection in humans after campylobacteriosis, and one of the leading causes of foodborne outbreaks. In 2019, almost 88,000 confirmed cases of salmonellosis were reported, with a notification rate of 20 cases per 100,000 inhabitants, the same level as the previous year. The disease trend has been stable for the past 5 years after a long period of downward trend. That the causes of this we will see a bit later. We can see in this graph that we have here on the right, where we have the number of cases versus the month and the year, and we see in this red line a clear stable average for these 5 years. The trend in human cases of Salmonella enteritidis has also stabilized. In 2019, 23 member states reported more than 900 outbreaks of foodborne salmonellosis, in which in turn caused just over 9,000 illnesses, almost 2,000 hospitalizations, and 7 deaths. This bacterium caused almost 18% of all of foodborne outbreaks in that year. The vast majority were caused by Salmonella enteritidis. The main transmission vehicle involved were eggs and egg products, followed by bakery products, pork meat and its products, and mixed food, which this refers to prepared foods, such as, for example, hummus. This was the same as in 2018. So to better contextualize, let's look at the few years outside of the EU, where the CDC estimates that Salmonella causes more than 1 million illnesses, 26,000 hospitalizations, and 420 deaths annually in the United States, with food again being the main source. In both the European Union and the US, Salmonella enteritidis and typhi are the most frequently reported serotypes. So according to Directive 2003/99, EC on zoonosis, member states must report on sources and trends, zoonotic agents, and foodborne outbreaks. Here in Spain, the Salmonella report is mandatory. To do this, the EU has adopted an integrated approach in relation to food safety known as One Health, and it is basically controlling Salmonella in the food chain from field to fork. It includes both risk assessment and the adoption of risk management measures, in which key actors such as the member states, the European Commission, the European Parliament, the EFSA, and the ECDC must participate. Control programs were implemented in each member state, for the reduction of Salmonella in poultry, which are, for example, laying hens, broilers, and turkeys. This added to restrictions on trade in products from infected animals. The EFSA provides independent scientific advice and support through the collection, analysis, evaluation, and recommendations related to Salmonella food safety to protect consumers. These monitoring programs are one of the most important milestones in the EU. And this approach has contributed to what we've seen before, which is the reduction of salmonellosis cases by almost half between 2005 and 2009, in part thanks to the making of the bacteria a priority. So before 2004, more than 2,000 cases were reported in 15 member states, and this was reduced to 90,000 cases, more than half, per year in 28 member states, meaning the double. Regulation 2003/99 also requires the absence of Salmonella in 25 grams or of almost every product, including carcasses and ready to eat foods. So there is, of course, a microbiological criteria for various food, which we will see next. And it is important to mention that all actions have also been taken to sensitize consumers, because an increasing consumer concern about food safety and contamination by pathogens has been recognized. So we can define a food safety criteria as the acceptability of a product or a batch of food products that are is applicable to products that are in the market, and this can be based on a microbiological criteria. In this slide, we have the microbiological criteria for Salmonella that must be met by type of food. They are not all, of course, but some, just to give us an idea. We can observe that in general, there must be an absence in 25 grams of sample. In addition, it is also possible to indicate the number of units of which the sample is composed. Here we see n equals 50, n equals 5, n equals 30. But again, we see 50 and 5 repeated multiple times. And here highlighted are the latest microbiological criteria that have been added, such as pulp, juice, and cocoa concentrate, reptile meat, raw milk for direct sale to consumers, and for pets, raw food, and chew accessories. So in general, we could say that a satisfactory result would mean an absence of Salmonella in 25 grams per sample. So to reduce this spread of Salmonella, again, the EU cracked down on the introduction of extended control programs and legislation covering routes of exposure to Salmonella. As we've seen previously, the absence of Salmonella is required in various types of food, mollusk, poultry, dairy, meat, eggs, of course, also fresh and ready-to-eat foods. And furthermore, this absence is evidence and supports both HACCP control and good industry practices. So one goal has been for each member state to reduce Salmonella in poultry by 10 to 40%, depending on their numbers in the previous year. And a reduction of 2% per year must be achieved. One other regulation would maybe refer to sampling and the frequency with which it must be carried out, or maybe specific serotypes of Salmonella in certain hosts. And well, all of these are controls that start from production on farms until it reaches the table of consumers, and are a systematic set of processes. For this part, the World Health Organization gave a couple of years ago recommendations for Salmonella throughout the food chain, which aim to both strengthen food safety standards by monitoring Salmonella, as well as educate consumers and train handlers in best practices to prevent this and other foodborne illnesses. Additionally, the WHO also highlighted the importance of both national and regional surveillance networks to stop the spread of Salmonella, as a contaminated batch can pass between country barriers, as we've seen in many outbreaks. So we have already reviewed the context behind Salmonella a bit as a primarily foodborne pathogen. So we will get now into the review of the ISO method. ISO 6579 is made up of 3 parts: detection, enumeration by most probable number, and guidelines for serotyping. This time we're going to review part 1 of 2017, including Amendment 1 of 2020, which mainly refers to a wider ranging incubation temperatures. So this ISO method for the detection of Salmonella is applicable to products for both human and animal consumption, environmental sample from the feed production and handling area, and sample from the primary stage of production. We will review the first, which are, of course, products for human animals consumption and environmental samples. Going fully into the method, it is very important to mention that Salmonella can usually be accompanied by enterobacter acid or other type of bacteria. So we have to have in a pre-enrichment stage to allow detection even when there is Salmonella in small quantities or injured. For this step, we use buffer peptone water, 25 grams of the sample mixed with 125 milliliters of buffer peptone water to obtain a dilution, and this is incubated between 34 and 38 Celsius for 18 hours. For the next step, which is the enrichment in selective media, we can use Rappaport soy bacillus broth RBS or MRSB medium, and this in parallel with the MKTTN medium. So 0.1 milliliters of the culture obtained during the pre-enrichment are inoculated in either 10 milliliters of RBS broth or in MSRB agar, and on the other hand, 1 milliliter of the same culture is inoculated in 10 milliliters of MKTTN medium. They are incubated at 41 and between 24 and 38 Celsius respectively for 24 hours. In samples of powdered dairy products and cheese, Salmonella may have been severely injured. So it can be incubated in this selective media for an additional 3 hours. Afterwards, we have the isolation in a selective solid media where both XLD agar and a second complementary medium must be used. So for this, we recommend Condachrome Salmonella because it is based on different agents from those of XLD. However, other media such as Salmonella Shigella agar, Wilson Blair, and Ectoan enteric agar can also be used. For RBS and MKTTN broths, a 10 microliter loop is used for inoculation, while in the case of MSRB plates, a 1 microliter loop is used to inoculate the selected media plates. The XLD agar is incubated between 24 and 38 Celsius, while the Condachrome Salmonella at 35 Celsius, both for 24 hours. The chromogenic can be even less for 18 hours. So in the XLD, typical Salmonella colonies have a black center and a slightly transparent reddish area due to the change of the indicator. In Condachrome Salmonella, the colonies will be a magenta. Although there may be other enterobacter acid that may accompany it, which would grow colorless, even or even blue-green, like Escherichia coli. For these species colonies, a subculture is carried out in a non-selective medium, such as a nutritive agar, just to have well-isolated colonies and to carry out the confirmation through biochemical and serological tests. We incubate this non-selective medium at 34 to 38 Celsius for 24 hours. In biochemical tests, it is mandatory to perform at least the culture on TSI agar, urea agar, and the medium for the decarbolization of lysine. while the detection of beta-galactosidase and the indel test are optional. For the serological test, the elimination of auto-agglutinable strains is carried out, since this would complicate the following tests, which are to examine the presence of OH and optionally B antigens. So in this slide, we have quickly what would be the method for samples from the primary stage of production. It is exactly the same as the previous one. The only thing that changes in this case is in the selective enrichment step, since only the SRB agar is used. This is because, given the nature of the samples, it is understood that the recovery of Salmonella should be way easier. So now, how are the results obtained interpreted? In this table on the left, we have the results for Salmonella typhi and other strains such as Salmonella enteritidis. The reaction that the first would have is shown, and the percentage indicates that there are even variations between and within the same serobar. That is, there may be difference, although very limited, within the same Salmonella typhi. On the other table on the right, we would have the final interpretation of both biochemical and serological tests, and what would be considered a confirmed Salmonella, a presumptive Salmonella, or a bacterium not considered Salmonella. So confirmed Salmonella can be further serotyped, and for this we have part 3 of the same ISO 6579. In this slide, quickly, we can observe Salmonella colonies in the different media. Here we have on the left an XLD plate with typical Salmonella colonies with a black center. Here we have TSI agar with different bacteria, where mostly typical Salmonella cultures show the red slanted surface and yellow bottoms. And of course, finally, a plate of our Chondrochrome Salmonella with its perfectly magenta colored colonies that will be the only ones to consider. So we are going to comment very quickly on something exciting that we are going to launch in 2022, which is a new line from Conda on an alternative method that can be used in the analysis of pathogens in the food industry. As we all know, traditional methods are the gold standard in this industry. However, of course, they are very time-consuming and labor-intensive for the laboratory personnel. For this reason, every day the market demands alternative techniques that are like traditional methods, meaning reliable, sensitive, specific, but that also obtain results in less time. The available techniques that are currently implemented on the market to a greater or a lesser extent include molecular diagnosis, qPCR, which is really the most widely used technique, but there are also immunological tests such as ELISAs and others like MALDI-TOF or NGS that I'm sure they will have a greater push in the following years. In this sense, the PCR has been used for years in the food industry for a rapid detection of pathogens. This technique is based on amplifications to obtain a large number of copies of a specific DNA fragment present in a sample. There are different types of PCR, but the most common is real-time PCR, also known as RT-PCR or qPCR. So what's the difference between a PCR and a real-time PCR? Is that this later, the qPCR gives the possibility of detecting the state of the reaction by fluorescence, which can be achieved using different techniques, one of the most used being that of probes that have a higher specificity than with, for example, stains, and therefore negative results do not require confirmation. And for example, this would look as here on the image, a typical qPCR reading where the different colors would correspond to different bacterial species. And at first glance, here we can see very clearly the difference between the traditional method and the alternative method. But once again, following the ISO, we have several steps: the pre-enrichment, the selective enrichment, the isolation, and the confirmation that is made up of several tests. And we would have results within 5 to 7 days with a qPCR technique in general, we can say that an enrichment step is also necessary, but the next day the extraction can be carried out and the PCR run. That is, in 2 days, the results would be available, which is why it is clear that the main advantage of these alternative methods is that it gives us speed in obtaining results, but it is not the only one. The qPCR also provides high sensitivity and specificity to detect the pathogen. Not everything is perfect because, of course, there are also disadvantages that we find in this technique, that it could amplify any fragment to which the probes bind, even that of non-viable cells, which would give rise to possible false positives. And well, also there could be contamination of previous PCRs. So there is maybe a risk of false positives as well on that part. So how from CondaLab we address these complications with our new CondaGene product line. And this is by adding internal and additional controls to ensure protection against false negatives. That is, eliminating traces of previous amplifications and with which exclusively the amplification of viable cells is ensured. In addition, these are protocols based on standardized methods, so the accreditation would not be necessary. These are open for open systems and compatible with a large number of equipment on the market, and protocols that guarantee minimum manipulation for a very simple process. We will of course have a kit available for Salmonella and for other microorganisms of great microbiological interest. To end the webinar, we present the products that we have at this moment available. Our catalog includes all the media and diluents indicated in ISO 6579 that we have reviewed, and we have, of course, also the availability in a wide variety of formats, both in dehydrated and ready-to-use media to, well, avoid any preparation times. And of course, we have our trusty Condachrome line for a fast and reliable response. Just in time, we are wrapping up the webinar, so let's quickly answer some of the questions you've been sending to us. Okay. Okay, here we have any other species of Salmonella that causes salmonellosis? Well, all the non-typhoidal Salmonella causes salmonellosis. So just it's Salmonella typhi, and there are other 3, I believe, serotypes that cause the typhoid fever, but all the rest of the Salmonella are the ones that cause salmonellosis. Detection by ISO method, it's taking 4 to 5 days, in the best of cases, of course, and by PCR it takes less time. Which method is best? Well, I believe this depends on what you have available in your laboratory. I mean, if you have a high volume of samples and you are very interested in having the results in less than a week, I would definitely recommend the PCR. They are both good techniques, but again, it depends on the situation. And if you have a high volume of samples to process, I would say it would be best a PCR. Much less complicated, but again, you would have to have equipment and all of that. That it's, well, an investment, but a good investment, because it seems that is the market trend, getting further away from the media culture and going straight into new, quicker techniques. Okay. Are all the requirements for Salmonella waste absence in 25 grams? I want to go back to the table that we saw with the products a bit further here. So it depends on local regulations, if there is, but it does not always has to be an absence. In some foods, there is a permitted number of samples in which there may be a certain number of bacteria that must be within the threshold value and the limit value. So, for example, in the case of an absence, satisfactory result would be this absence. In any other case, a satisfactory result would be if all the observed values are fulfilled. For example, here we have very quickly bovine, ovine, goat, and equine carcasses. We have 50 units that make up the sample, and there are 2 units of the samples that are allowed to have to be between the threshold and limit values. Here they do not call for absence. So yeah, you would have to review your local or regulations or national regulations. Can Conachrome be used for enrichment steps for PCR? No, you would need because Conachrome is for an isolation, not for enrichment. So depending on the protocol, maybe it is simply Peptone water, just like in the ISO, and that's it. Or maybe it's a more selective one. So it depends on the protocol, but no, Conachrome is just for isolation. For the serotyping of Salmonella, okay, you can go to part 3 of this ISO 6579. There you have all the information. There are several steps to be taken, so you would have to go through that. The difference between PCR and advantages of qPCR we've already mentioned. I have worked with XLD, and the plates sometimes look weird, cloudy, when it is usually transparent. So it is very common that this happens with this medium, the XLD. And what you are observing are precipitates, and it usually happens when working with large volumes of this medium. It is due to overheating when dissolving the medium, since, well, at higher volumes, the medium remains exposed to higher temperatures, and the cooling is, of course, lower. So in our experience, you have to be very careful and dissolve quickly so that the medium does not suffer. So I think, can we proceed for qPCR from the pre-enrichment media? Yes, of course. Yes, that would be the method, the A enrichment, and then you can do the extraction of the DNA and run the PCR. Okay, so I'm going to end the webinar here. Let's see. So we hope this webinar has been very interesting for you, and well, both the recording and the certificate of participation, you should have it by tomorrow in your emails. We want to thank you again for joining us today at this Condalab talk. We hope it has been useful, and if you have, well, any other questions, you can write us at micro@condalab.com, and then I think our marketing department will send a very brief survey just regarding all the Conda Lab talks that we have made during this 2021. So we invite you to answer it, to improve with the subjects and the content. This always helps us. And we have very interesting things planned for the next year. One of them, which is Condagene. So do not forget to follow us on LinkedIn and subscribe to the newsletter, because that is the main place where you will find all the information regarding this launch. And at last but not least, I would really like to take this opportunity to wish you all, from all of us in Conda Lab, a very happy holidays and an excellent start to the year. So until next time,